So this topic will be a shorter one, aiming to cover just some basics of how neurons work and some basic stuff about the brain that'll be useful to refer to throughout the rest of the course. If it's already familiar to you, that's fine. I hope it's a helpful review and will clarify some details. We'll start by looking at a neuron. So here's a simple diagram, a simplified diagram of a neuron with a few parts relevant to our purposes. First off, neurons take in some sort of input, and the simple common kind of neuron we see here takes in input through its dendrites, the green part here that has a sort of tree-like branching. Now those dendrites then send their input, send their info to the cell body. This is often also called the soma. The root word soma just means body, and we'll actually use that in other contexts for this course, so it's worth pointing out here. The cell body is where the nucleus and all the organelles and stuff is, but the important thing for our purposes is just that the inside there, it's got an electrical charge. Specifically, when it's just sitting around resting without stimulation, a neuron is negative, around negative 70 millivolts of electrical potential. Okay, finally, if circumstances are right, the information will be sent from the soma down the axon, shown here in blue. Note that this isn't to scale. Neurons are actually super tiny, but the axon can be pretty damn long. Like a single neuron could connect areas of your brain pretty far apart, or out in our peripheral nervous system and the rest of our body, there are neurons that have an axon multiple feet long. A single cell in your body that stretches from your toe up to your spine. And the reason for the axon is that it's the output of a neuron. It's where the neuron will send information to something else, like the next neuron in the chain, or if it's a motor neuron, then a command to the muscle it's attached to. Note that at the end of the axon, you can see these little terminal buttons that kind of branch out. These are what actually send the info onward. Now, most of our neurons are not sensory receptors, like our, skin things that pick up touch, nor are they motor neurons, like the things that turn on muscles, but rather we have billions and billions of what are called interneurons, just meaning neurons that hook up to other neurons. So how do neurons hook up to each other? Like, how do they pass info from one neuron to the next? Well, like I mentioned, signals will go from the end of one neuron's axon at those terminal buttons, and that becomes input to the next neuron's dendrites. So it goes from the blue of one neuron to the green of the next neuron. But importantly, the neurons do this without actually touching. There's actually a tiny little salt-watery gap between them that's called the synapse, or the synaptic gap, or the synaptic cleft. So here I've kind of zoomed in on a synapse where the axon of neuron 1 almost meets the dendrite of neuron 2. So you can see in this little zoomed in thing here, the synaptic gap between them. So here would be the axon of the first neuron, what was blue over here. And here at the other end is part of the dendrite, right, the receiving part, so dendrite, of the next neuron, the second neuron, the next one over, okay? So if there's a gap there, the synapse, right, how does the first neuron get a signal to the second neuron? How do they communicate? Well, when a neuron fires, it sends an electrical signal down its axon to the terminal buttons. You can kind of see some drawing. The drawing here has some kind of electricity going down the axon in the upper right there. Again, that's the first neuron, the one that has fired some info down its axon, and we're seeing a terminal button here at the end, right up in that synaptic gap, that synapse, right next to the neighboring neuron's dendrite, okay? Now, when the electrical signal gets to the end of the axon, the terminal button actually releases neurotransmitters, which are chemical messengers. That's why we say neural communication is an electrochemical process, right? There's an electrical part and a chemical part. So here at the chemical part, these neurotransmitters, shown as the little kind of red spheres here, they float across the synapse area. And some of those end up fitting into receptors on the next neuron's dendrite. So there's a different meaning of receptor than what we've talked about so far. When I talked about the sensory detectors, right, the sensory neurons that we have, I call those sensory receptors, like the hair cells in our ear that pick up sounds of a certain frequency. Those are receptor neurons, receptor cells. Here we're zooming in on a little piece of a neuron, a little piece of one neuron. On the dendrite itself, on those little dendritic spines, we have little openings, little holes, and we also use the term receptor for those. So those are receptors to receive or hold on to or grab things like neurotransmitters. Now, there are actually lots of different neurotransmitters. You've probably heard of many of them, like serotonin and dopamine, epinephrine or norepinephrine, sometimes called noradrenaline. There's glutamate for excitatory processes. There's one called GABA for a lot of inhibitory processes. Acetylcholine is important for like memory and other stuff. Oxytocin, you've probably heard of, substance P, and a ton of others. All of these neurotransmitters, though, have different molecular shapes. And those tree-like branches on the dendrites of neurons have receptors that are specific to some neurotransmitter shapes, but not others. So really, neurotransmitters only fit into particular receptors. It's kind of like a lock and key mechanism. So a good metaphor for this would be like a lock and a key. You can only open a lock if you have the right key, and you can only open the receiving neuron on that dendrite if you squirted out the right type of neurotransmitters to bind into those receptors on its dendrites. So what happens when a key fits into a lock? Well, the lock opens. So when neurotransmitters fit into receptors, it causes an opening. It actually causes ion channels to open in the receiving neuron's dendrite. Remember, ions are just charged particles, things with electrical charge as either positive or negative. Neurons are kind of all about electricity. So what happens when these ion channels open up? Well, remember, I've mentioned before that in its normal resting state, when it's just chilling, nothing's going on, a neuron has a negative electrical charge. It's about negative 70 millivolts. So we say it's polarized. Polarized just meaning away from zero or away from neutral, right? Like if someone's politically polarized, it can be in either direction, but they're definitely not neutral. The more polarized they are, the further they are from neutral in either direction. So being either positive inside of a cell like this or negative inside would make that neuron polarized. In this case, since most neurons are normally sitting there waiting at negative 70 millivolts, they're sitting there polarized, okay? Now, when the ion channels open up, because a receptor got filled, right, it got unlocked, this ion channel lets in a bunch of positive ions, a bunch of plus signs, basically. Specifically, sodium, which would be Na plus, will flood in, and that positive means it's got a lot of positive charge. It'll make the inside of the neuron be less negative. So instead of negative 70 millivolts, it gets closer to zero. It gets less polarized. So we say flooding in all that sodium is depolarizing the neuron. And if enough positive charge floods in, enough of those pluses come into the normally negative cell, if enough depolarizing occurs, in other words, if the neuron becomes not so extremely negative, then that receiving cell will fire. That's what makes a neuron go off. So here's a picture to make that a little more clear. On the y-axis to the left, we've got the electrical potential, the charge from positive to negative. You can see the neuron when it's sitting there in its resting state is around negative 70. But then if it gets some input from its neighbor neurons, if enough of its receptors get filled with the right neurotransmitters, then it depolarizes. It gets less negative, right? It's going back towards zero or even towards the positive. At some point, if it reaches a threshold, a sort of cutoff, which is usually or often around negative 35 or negative 40 millivolts, it starts a runaway chain reaction process that makes the neuron fire. You can see it shoot up to a positive charge here. That's the neuron firing. And then it goes back down to its resting state. So, through some ion pumps, we won't worry too much about the details of those mechanisms, but it gets it back down to negative. It even overshoots it a little bit and briefly goes below negative 70, but it'll quickly end up back at negative 70 in its resting state, ready to fire again to new input. Now, I showed it before as one neuron connected linearly to the next neuron and that one to the next, all in a nice straight line. But for the most part, especially in our brain, neurons tend to be connected to tons of other neurons. One neuron can potentially hook up to as many as 10,000 other neurons. On average, a neuron can have more than 1,000 synaptic connections. So we've got somewhere around 100 billion neurons in the brain, which is a ton, but something like 100 trillion connections in our brain. So really, you've got to think of the dendrites of a neuron as getting input from a buttload of other neighboring neurons, not just one neighboring neuron. And if we want to know whether a given neuron will fire, like this one I've pointed to here, it actually depends on the combined input going into all of its dendrites. In other words, does the total net charge become positive enough, right? Depolarized enough, or not as negative inside. And for each of those earlier neurons, they've also got a ton of other neurons connecting to them at a ton of little synapses. So what we've really got is this super complex orchestra of activity constantly going on, and each neuron is doing some simple math, like adding up its inputs, like plural, multiple inputs, and only firing if it gets enough total input. But the reason I talk about net charge, not just adding it up, is because sometimes the input from one neuron is negative. The input from one neuron to another, we say, is inhibitory, meaning that if that connection activates, if that synapse goes off, or the receptors get their little neurotransmitter inside them and open up the ion channel, if that dendrite's receptor gets opened by its neighbor, it actually makes the receiving neuron more negative. We would call that inhibitory input, or an inhibitory connection. I've drawn kind of minus signs here. Other connections, of course, and probably most of them, more of them, are excitatory connections, where activating it makes the receiving neuron more positive. So I've drawn plus signs here, which again means that it'll help depolarize the neuron. It's not as negative as its resting state was, so these make the neuron more likely to fire, right? More likely to get up to that threshold of negative 35 or negative 40, where it'll start the chain reaction and fire. So adding up all those pluses and minuses, the excitatory and the inhibitory inputs, the inside of the neuron has to reach a threshold, a cutoff, like I said, around negative 35 or negative 40, in order to set off that chain reaction. And when it does fire, we call that an action potential. An action potential means it fires an electrical signal down the axon. So here, kind of going to the right on the blue here, fires it down the axon, and this travels really fast. It can go even faster if the neuron is myelinated, meaning it's covered with something called myelin sheath, just an insulating layer that helps the electricity travel more efficiently. But either way, regardless, we won't worry too much about that. Either way, we call it an action potential, just meaning the neuron has fired. It's a bit of a weird term, but just remember the term potential here comes from electrical potential. It's an electrical process at this stage. Now, the action potential travels down the axon, and it's at those terminal buttons at the end where the electrical signal causes them to open and release neurotransmitter chemicals, becoming a chemical process to transmit the info to the next neuron. Here's kind of just a diagram to make this whole thing more clear. Again, we're just graphing out the electrical potential inside the neuron, like how positive or negative it is. So you can see the y-axis going from negative 70 up to 0 here. Of course, it could go positive as well. In the upper left here, we've got a neuron that's resting at negative 70 millivolts, and then it suddenly gets some excitatory input. In other words, stuff like sodium, right, Na plus, floods in, makes it more positive inside. But in this case, in this example, it wasn't enough to reach that threshold. It didn't get depolarized enough, so there's no action potential. The neuron doesn't fire. Not like it got some input, but it doesn't pass along the signal. Like someone said, pass it on, but they said it so weakly that it didn't actually pass it on. And soon that neuron just goes back to its starting point, chilling at negative 70. In the upper right here, you can see what happens if there's more excitatory input, though, if there's enough. Like once it reaches the threshold, the dotted line, you don't even need more input from neighboring neurons. At that threshold, if you get there, a chain reaction just takes over, and the neuron is going to fire. It zaps up, it shoots the electricity down its axon, and then the inside of the neuron goes back to negative 70 soon afterwards. This happens really fast. It can happen like 100 times per second a neuron can fire, or even more, which we would call that 100 hertz, right? Something happening that many times per second is hertz. Though if you average across all the neurons, it averages out to more on the order of maybe a couple times per second on average for neurons. Like there are some quieter neurons that may even go seconds or potentially even minutes without firing. Although again, others may be almost constantly firing, 100 times per second or more. Finally, in the bottom example here, what we've got is an example where the input to the neuron is more inhibitory than it is excitatory. And in that case, the neuron gets even more polarized. We could even call it hyperpolarized, and it'd be even harder to get this one to fire until it gets back to at least its resting state. Now, Importantly, if there's not enough total positive input to reach the threshold for an action potential, then nothing at all happens. The cell does not fire slightly or fire weakly. Action potentials are all or nothing. They either happen or they don't. So you might wonder, how does the nervous system tell the difference between minor stimulation versus major stimulation? Like how your touch receptors can tell the difference between a light touch and a heavy touch. The way that it's encoded is not by an individual neuron firing more strongly or more weakly. Even single action potentials, every single action potential, they look the same whether we got way over the threshold or just barely over that threshold. Every action potential is identical. So the way a stronger stimulus is encoded is actually by more neurons firing, and or the neurons are just firing more often. They're firing quicker. It's not that it's a stronger firing. Each individual action potential is always the same strength, but maybe more of them are going off or they're going off quicker. An analogy might be like a gun. Your finger might pull the trigger just a tiny bit back. Pull the trigger back just a tiny bit, but that might not be enough to fire it, right? You pull it back a tiny bit, but if that's not enough, the gun won't fire. You didn't reach the threshold to make the gun go off. But if you pull the trigger enough to fire the gun, it doesn't matter if you just barely pulled it to that point or slammed the trigger back with a ton of force. In either case, the gun just fires. It fires the same. It doesn't fire weakly, right? It doesn't fire just a little bit. The bullet doesn't come out faster because you pulled the trigger harder. It either fires or it doesn't. That's what we mean by all or nothing. That's how action potentials are. That's how neurons work. So just for example, if you were to hook up an electrode, a little measuring device, an electrical measuring device, hook it up to a single touch receptor neuron, just meaning like a neuron there in our skin that picks up touch. So hook it up to that neuron, to the axon coming out of it, and count the number of action potentials under 3 situations, like a light touch to that area where the receptor is, or a medium touch, or a strong touch. You would see something like this. You'll actually often see us draw out action potentials like this. You'll see a lot of diagrams that look like this to give a feel for how quickly a given neuron is firing. Now note in the representation here how a stronger stimulus, like meaning more pressure on the skin, makes this touch sensing neuron fire much more often, whereas the light touch means not as often. So the light touch, the neuron's going pew 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 pew, whereas the strong stimulus means that same exact neuron might be going pew 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 pew, like firing really fast. Now note how in the representation here, what we can see in this diagram, a stronger stimulus, more pressure on the skin, it might make it fire more often, but every action potential is the same strength. So we've drawn the bars at the same height here just to really emphasize that. Now here's just another representation of this whole idea that actually shows a really important detail we want to make note of. Neurons are rarely just sitting there without firing. Neurons actually have a baseline rate of firing, even without any stimulus, kind of just some random firing once in a while to keep the neuron alive and kicking. So a stimulus, like touch to the touch receptor neuron, a stimulus will make that neuron fire more often, more than its baseline level. Or I guess if it's an inhibitory connection, right, it could even be less often for inhibitory input. But each firing of the neuron is the same action potential, as we can see at the top here, where we've labeled spikes. We just mean action potentials, what I've been doing like the pew 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 thing, right? So notice how when there's no stimulus at all, right? Early on here, there's no stimulus. The neuron is firing. It's still doing its like occasional baseline firing. It's going pew, 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 right? But that's just like the neuron keeping itself alive. It doesn't tell us anything about stuff going on out there in the world. However, if we get over that threshold, like in blue here, right? If we get enough input, then the rate goes up. Then it's firing pew, 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 pew. It's a little more. If we just got a little bit of stimulus, like in the red here, we got some stimulus, but not enough, right? Then nothing really changes. It's still just doing its baseline rate of firing. It's pretty similar to back here. It's every once in a while, pew, 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 right? It takes enough to get over that threshold, and then what's happening when we say, we got enough stimulus, enough stimulation to make the neuron fire, what we really mean is make it fire more action potentials than its baseline rate, to make it fire higher than baseline rate. That's what it means to get stimulated enough for that neuron to react, right, to say we've really detected something. And then again, if we get a much stronger stimulus, like in green here, go way over the threshold, a strong input, a strong stimulus, then the firing rate may go way up. It'll keep firing quickly, all in a row, pew, 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 like really fast. And that's how the brain will be able to tell the difference from that single wire going to the brain, from this one little touch receptor, say. That single wire, the brain will know whether it was stimulated a lot or just a little bit, based on if it's firing a little bit above its baseline rate or a lot above its baseline rate. Finally, one last diagram I wanted to show just to bring this stuff together. Here you can see we've labeled 3 of the synapses, the little connections on this particular neuron's dendrite. So we've got the neuron here with its cell body, and you can see the tree-like branching coming off. These are all dendrites, right? It's labeled here dendrites. But on those dendrites, you got the little connectors, the little places where it hooks up to a different neuron's axon. So this is some other neuron's little axon coming down to a terminal button here. Here's maybe a different neuron hooking up with its terminal button here, and maybe a third other neuron hooking up to this one at the synapse here. Okay? And we've labeled these 3 incoming connections from other neurons. 2 of these are excitatory, so we've labeled them E1 for excitatory 1 and E2. While the other is inhibitory, so labeled I for an inhibitory connection. If that one's activated, it can actually flood in stuff like Cl-. So with the minus sign, means it's a negatively charged ion. That would be like chlorine ions flooding in, making it more negative inside. So if this inhibitory synapse, turns on, connects here and activates, opens ion channels here, what might be flooding in is actually negative signs, making it less likely to fire here, right? Hyperpolarizing it. So here at the bottom are 4 situations and what the neuron would do, so you can see how a neuron is kind of doing some math, some addition and subtraction inside the neuron to decide whether to fire. So in the first situation here on the left, right in the bottom left here, some activity happens at, let's say, the E1 connection or maybe at the E2 connection, but that might not be enough to reach the threshold. Now in the second one here, the second example, if both E1 and E2 connections are active, that may be enough to reach the threshold, in which case we get an action potential. In the third situation here, you see the inhibitory synapse I, that was activated, so it actually hyperpolarizes. Obviously, no action potential there. It makes it even harder to fire. It's nowhere near the threshold. And then in the fourth situation, this is a more interesting one, all 3 of the synapses have had some activity. And in dotted lines, you can see the total positive input dotted with blue lines. That would be from E1 and E2, but also the negative input dotted red. And what they've drawn in orange here is the net effect, kind of adding the combined effect, the pluses and minuses together, which you can see in this case didn't reach the threshold here. Even though it got just as much from E1 and E2, it was kind of canceled out by the connection at that inhibitory place. So in this case, no action potential. All right, now let's put neurons into context within the body. Our entire nervous system is just a big system of neurons throughout our body. When we talk about nerves or nerve cells, those just mean neurons. That's all that means. So it's a system of neurons throughout our body, and it's really broken down into 2 major parts, 2 major subsystems. First, we have what's called the peripheral nervous system. So here in red, and you can see throughout the body here in red. The peripheral nervous system is all the stuff throughout our arms and legs and skin and stomach and basically everything that isn't our brain or spinal cord. The peripheral nervous system includes, we'd include under a subset here, the somatic nervous system. That would include sensory neurons, that is what we've called sensory receptors, like touch receptors in our skin and the little hair cell receptors in our cochlea. Those send info into the brain, like when we're touched, the touch receptors in our skin sends the info to the brain. And this also includes the peripheral here, and the somatic part of it also includes motor stuff, motor neurons, where the brain would send a signal out to our muscles to activate them. It also includes autonomic stuff, kind of automated reaction stuff, like the 5 Fs: fight or flight, freezing, feeding, and mating. Now, our brain and spinal cord, on the other hand, here in green on the right side, make up what we call the central nervous system. If you're ever reading about an explanation for a neurological phenomenon, like say tinnitus, and you've read it described as being central in origin, they're saying it's due to activity in the brain, in the central nervous system, right? Brain or spinal cord, usually the brain. If someone is blind, for example, They can be blind due to problems with their eyes, which would be a peripheral cause of blindness, or they could be blind due to the visual part of the brain, the visual cortex being damaged. And in that case, that cortical blindness, we actually say it has a central cause. You might read of it as a central visual deficit, and they're just talking about it being due to some issues in the central nervous system. Regardless, whether it's the peripheral or central nervous system, it's all made up of neurons, like these little neurons we've been talking about. So all throughout the body, all throughout the brain and spinal cord, just a big old bundle of tons of neurons. And of course, there are other support cells and things that go along with it, but it's just a bunch of neurons make up the nervous system. Now, we've obviously got to communicate between the central and peripheral nervous system. They're not totally separate. Our brain needs to get input from our sensory receptors, but it also needs to send commands out to our motor system so we can move our muscles around. So it's all connected. Now, when we're talking about signals going to the brain, like sensory signals bringing information from the peripheral toward the central nervous system, to the spine and then the brain, we call those afferent signals with an A. Afferent signals or afferent pathways. Whereas efferent signals with an E, or the efferent pathway, refers to motor signals, meaning like we're sending information from the brain out toward the peripheral nervous system, like out to muscles. Okay? Now, it's unfortunate that these terms sound so similar, so you may need a mnemonic to remember them. I like to think of it as afferent signals with an A are for things that affect me. I am affected by the world through my senses, right? Those words start with A. Whereas efferent signals with an E is me acting on the world, being effective. I am having an effect on the world through my motor actions and movements. Those start with an E. That might be one way to remember it. Other mnemonics I've heard would be afferent arrives, efferent exits the brain. So A with A, E with E. Afferent arrives at the brain, efferent exits the brain. That's how you might tell those apart, but now we'll be able to use those terms throughout the semester and refer to afferent connections or afferent signals or pathways. That just means like we've picked up some sensory information and sent it to the brain, or efferent signals like those outgoing commands to like our motor system. All right, enough at the level of neurons. Let's dive into the brain in our next video.